This is Professor Ralf Hausmann from the University of Aachen. And he's a group leader at the clinical pharmacology. And he was one of the former grant winners of the SyncroPatch grant. And he's interested in molecular aspects of ligand binding and understanding mutations in sodium channels that are associated with pain. So I'm very much looking forward to your talk, Ralf. And stage is yours.
Thanks for this kind introduction. And also thanks for inviting me to this really exceptional meeting. Today, I would like to talk about the drug screening, which reveals inhibitors of sodium channels and their variants. And it's not working.
The cursor has to be.
Sorry for that. The work was done in the framework of the so-called Sodium Channel Network Aachen, and this is a research consortium from scientists from the fields of clinical pain research, genetics, electrophysiology, pharmacology like me, computer modeling, and also functional imaging by MRI, and also preclinical research using animal models of pain. If you're more interested in this faculty-funded project, please visit our website. It's available here. And today I will mainly focus on the sodium channel 1.7 and its role in pain. The role of the NaV 1.7 in pain is obvious from loss-of-function mutations, and this can cause the chronic insensitivity to pain. And most of you will know these people who can run on red burning colds. Also, gain-of-function mutations are known for the sodium channel 1.7, and these result in pain syndromes, for instance, like the human inherited pain syndrome, the erythromelalgia, the paroxysmal extreme pain disorder, PEPD, or small fiber neuropathy, SFN. And today I will show you the PEPD, or a novel PEPD mutant, the paroxysmal extreme pain disorder. This is an autosomal dominant chronic pain disorder, and it's usually a gain-of-function mutation in the sodium channel 1.7. The clinical symptoms are really the horrible pain attacks with intensive burning pain in the rectum, eye, jaw, different regions of the body, and these are accompanied by a flushing in this harlequin-like presentations like shown here. And on the left, you see a schematic of the sodium channel topology with the 4 domains. And this is taken from a recent 2019 review of the Waxman lab. And here are shown some mutants that are already known to cause this PEPD. And most of them are located intracellularly and mainly in the linker region. For instance, here in domain 4, the S4, S5 linker is shown here. And there was also one mutant which was presented by Angelika 2 years ago on this meeting here. And this was a picture from the initial publication of this pain syndrome. And what's the biophysical phenotype that is known for these mutations? And this I show you with this example already presented by Angelika. And you can see that we have, due to the localization of most of the mutants, we have an impaired inactivation of the sodium channel, and this results in a increased persistent current, which you can also see from the current traces from manual patch clamp that are shown here. And now we have identified in our research consortium a novel PEPD mutation in the sodium channel 1.7. It's the methionine 1628, which is mutated to a lysine residue in this channel. And these are 2 patients with the typical PEPD symptoms in a German pain center. And in these 2 patients, the mother and the daughter are affected, and sequencing reveals the mutation in the SCN9A gene, which causes the mutation I mentioned before. If you look where it's located, and here's again the sodium channel topology, and you can see it's directly adjacent to an already known mutation, the 1627 to lysine mutation in the typical MM motif of the S4F5 linker in domain 4. And here in zoom in, you can easily see that this region here are the both methionine residues. This is very close to the IFM motif responsible for the allosteric inactivation mechanism of sodium channels. I will show you later some structural implications or some structural models what the mutants can cause. First, we did biophysics of this mutation in comparison to the already known 1627 to lysine mutant, and in comparison to the wild type 1.7 channel. This was done in the beginning by manual patch clamp, and then we had the device from Nanion, and it was also verified by the SyncroPatch device. And as you can see here, the activation curves here, the comparison of the wild type and both mutants, you can see the activation curves are completely similar, and there's no change in the V half of activation. And by contrast, if we look to fast inactivation of the channel, here in black, the curve of the wild type, and for both mutants, for the novel mutant 1628K and as well as the known mutant, we have a depolarizing right shift of the fast inactivation curve, which results also in this V half, which is more depolarized as compared to the wild type. And that means we have the already known typical biophysical property of a gain of function mutation. And yeah, this is in agreement with the literature for the PEPD biophysics, I will call it. Yeah, if we look here more closely to the structure, we have, as I mentioned already, we have the typical MM motif, which is unique to the domain 4 of the sodium channel. In all other domains, there are glycine or glutamine residues. And here we have this motif, and this is mutated in these patients having this thing. This is a view that you already have seen. If we have a closer look to this region, we have the 1627 and adjacent to it 1628, the methionine very close to the IFM motif. And what happens when we have the mutation in form of a lysine residue in this position, and the larger side chain containing the positive charge of the lysine residue, as shown here in the red spheres. And we propose that there may be much more steric clashes, which may impede the allosteric inactivation mechanism driven by the IFM motif here. Yeah, what did we now with drug screening? We asked the question, do approved drugs affect this mutation? We have this question because this may result in drug repurposing in special patients. And for that, we used the Pharmacon library from Microsoft Discovery Systems, and it contains APIs of approximately 1,760 approved drugs. And the first screening was done at a concentration of 10 micromolar. This is a relative high concentration, we know. And for that, we used the Nanion SyncroPatch 384 device, which we had from the SyncroPatch grant. And this was mentioned by Niels yesterday. Really, if you did not so far, you should really apply for it. It's a great opportunity to really dive deep into high throughput screening, and it's a great opportunity in academic research. I think it was a great time with this machine in our lab. As shown here, we used simple pulse protocols in intervals of 10 seconds, and then we compared the current after compound application to the one before compound application. And also beside the tonic block, we have analyzed the use-dependent block at two frequencies, 10 Hertz and 50 Hertz with this activation trains. And in between, we had also the compound application, and then could calculate the amount of inhibition of the different compounds. Here I show you a first example for the antiarrhythmic and calcium channel blocker diltiazem shown here, and you can see the use-dependent block at 10 and 50 hertz, and you can see that the wild-type channel is blocked or partially blocked or inhibited to roughly 20% by high concentrations of diltiazem. And if we look to the mutant channel, we do not see such an inhibition at these concentrations of 30 and 100 micromolar. Yeah, here I show you some more results of our screening. It's not a campaign that would be run maybe in industry with lots of hundred thousands or millions of compounds. We had a smaller number, but anyhow, these are approved drugs. And for instance, we had a kind of positive control. The first generation antihistaminic drug acylastine hydrochloride showed the known sodium channel blocking behavior. And then we had, as you can see here from this graph, most of the compounds did not, at the concentration of 10 micromolar, did not affect the sodium channel amplitudes in this. This is from the pulse protocols, from the tonic block experiments. But some compounds were found that showed a little stronger inhibition of the mutated channel as compared to the wild type, for example, butambane. This is an ingredient from a local anesthetic preparation for the mouth, for instance, or the very old antipsychotic chlorpromazine was shown here. And there were also compounds like the old tricyclic antidepressant amoxapine, which is more potent or more inhibitory, shows a more inhibitory effect at the wild type as compared to the mutant channel. And this was also true for the bromperidol. This is then we did, after this first hits, after the analysis of the hits, we did concentration response analysis with a 6-point dose response or concentration response curves. And here you can see, for instance, the amoxapine, which inhibits the wild type more stronger than the novel mutant, the M1628 to lysine. Clearly, if you look here now to the concentrations, we have it in the low micromolar range. This is in the region of 8 to 10 micromolar, for instance, like the amoxapine. And you have to take into consideration that indeed this is larger as the plasma concentration for most of these compounds. That's totally clear. But if you think for these mutants or for the patients, for instance, the carbamazepine was also shown to work only in the range of small micromolar concentrations. However, most of the patients, and for instance, the 2 patients who have this novel mutation, they go very well with very low low dosed carbamazepine, and that may show you that we do not need a full block of this receptor mutant, maybe low concentrations or lower concentrations, and a smaller inhibition may be already sufficient to get relief of the symptoms in these patients. Yeah, and this is also summarized here. I think most of the data you have seen. And here we have the positive control, the amoxapine bromperidol, which was shown here. What will we do in the future with this? We will increase the number of compounds. For instance, we will use virtual screening together with colleagues from the Forschungszentrum Jülich. And for instance, do virtual screening with large compound libraries, Sync database, other compounds and precursor libraries. And this will be done in combination also with the wet lab screening campaigns. For sure, we will use again the Pharmacon library, because the idea is this may enable in the future repurposing of drugs. And there we have another recent running project, recently funded and currently running project. It's funded by the Federal Ministry of Education and Research, the so-called Precision to Treat project. We have a large database in our sodium channel consortium in Aachen of a few hundred patients having neuropathic pain. And there are lots of patients who have some mutations in sodium channels, for instance, and other ion channels. Lots of patients have very common mutations, and only a few patients have very rare mutations. And the idea is that a special combination of also very common mutations can have a special phenotype. And therefore, the idea is to take blood from these patients, reprogram induced pluripotent stem cells, and these iPSCs will, in our lab, then differentiate it into patient sensory neurons, which then undergo APC electrophysiology with the pharmacon library. And in the future, because this may be a regulated study or maybe an in vitro diagnostic study, may result in precise personalized drug treatment of these patients. Okay, and at the end, I would like to mention this really nice seminar, which will take place in the end of January next year to beginning of February in Grindelwald. And it's a Worldwide Sodium Channel Conference. It's the first one. It's a follow-up of the very successful sodium channel seminars. You can have a look to the website, sodium channels seminars.org. And if you look to the list of speakers shown here, you can easily see this is really the who is who in sodium channel research. And you are invited to apply for this meeting. And it's really nice that also the Journal of General Physiology agreed to run a special issue, which is open until August, also independent from this meeting, for voltage-gated sodium channel papers. And for the people from industry, maybe some sponsors are welcome. And for instance, Nanion already has agreed to be a sponsor of this nice meeting. Check it on the internet page. And at the end, I would like to thank the group of Angelika, especially the medical student Eileen, who did most of the work, and the funding, the Faculty of Medicine of our university, and also Nanion for giving us the opportunity to use this device for at the end for more than 1 year, and it was a really fruitful collaboration. This paper was already mentioned by Niels yesterday, together with Martin from Nanion. We were able to do this temperature experiments, which would never been able with such a number of experiments with manual patch clamp and also other pharmacology experiments, but that they were already reported by Angelika 2 years ago. For example, 1.5 sodium channel or hERG pharmacology. Okay, thank you. And if you have questions, let me know.
Thank you very much, Ralf, for this great talk. Questions from the audience? And maybe I have one small question. So the 1628K, how would that affect the electrophysiology in the patient? So is the threshold for action potential excitation increased in those patients?
For sure, if you would go for some DRG recording of the patients, which is not possible, clearly, but you would see the current traces like we have shown. Whether this will result in a change in excitation, this is hard to answer, because maybe most of you will know the recent paper from I think it was the Prescott group, who showed that when you have in DRGs, for instance, when you lose the NAF 1.7, the excitability remains stable because it can be recovered or rescued by the NAF 1.8, for instance. And after a few days, they show that the excitability was unchanged due to the presence of both sodium channels, for instance. Therefore, it's really hard to answer this question, and we do not precisely know.
Thank you, Ralf.
Okay. Thank you.
There's one more question, okay, but I think we have to move on a bit.
Why do you think that the positive charge of the lysin is not important for the effect of the mutation? Or you mentioned it's the size.
This we don't know precisely. There were done some molecular dynamics analysis and simulation analysis, and we for sure think there may be one issue with the size and the steric clashes, but for sure the charge may be also important because we have shown for this mutant, which is also very close, the 1632, and there was a mutation from alanine to glutamic acid. And there was a great issue with the allosteric inactivation mechanism. And we saw that the charges are for sure of importance. It's not only the size. Yeah. I cannot prove it with data, but if you look to our experience with this and in this region of this allosteric inactivation site, then for sure the charge will have some impact on that.
Thanks again, Ralf.